binds with a receptor protein, a signal transduction pathway is triggered within the cell. Ultimately, the signal transduction pathway will result in a cellular response. The details of each pathway will vary depending on the type of cell signaling. Phosphorylation is key in the control of protein function. Phosphorylation is the chemical process of adding a phosphate group to an organic molecule, such as a protein. When a molecule is phosphorylated, it is activated or turned on. However, if the normal state is in an active form, then adding a phosphate group can turn it off. So phosphorylation changes the activity of a molecule. But just as you don't always want to have your lights on, a cell doesn't always want to have a molecule be in an active state. To get back to the original state, we have to undo the activation. In this case, the phosphate group needs to be removed from the molecule, and this process is called dephosphorylation. We'll discuss enzymes in much more detail in a different chapter, but for now, we need to know that enzymes are proteins that catalyze chemical reactions. That just means that enzymes speed up a chemical reaction.
Must go faster.
A lot of the time, but not always, the names of enzymes end with a suffix "ase," and sometimes the first part of the name of the enzyme gives you a hint as to its job. The class of enzymes that catalyzes the addition of a phosphate group, that is, phosphorylation, is called protein kinase. It ends with a suffix "ase," so that's a hint that this is an enzyme, and the first part, kin, means movement. And if you've heard of kinetic energy, then you've seen this root word before. Kinases move phosphate groups onto other molecules. The class of enzymes that catalyzes the removal of a phosphate group, that is dephosphorylation, is called protein phosphatase. Let's look at this name. The last name of this molecule is ase, so we're going to think enzyme. And the first part of phosphate sounds like phosphate. So we'll remember that phosphatases remove phosphate groups. Both protein kinases and protein phosphatases are important players in managing the signal transduction pathway. Let's look at this general representation. The ligand binds with a receptor protein. This results in the G protein, more details on this guy later, is phosphorylated using kinase. This extra phosphate group moves the G protein to another enzyme. The G protein is then dephosphorylated by phosphatase. The removed phosphate group is hooked onto the other enzyme, which will then activate a secondary messenger. Secondary messengers are used in some signal transduction pathways. The job of these molecules is to amplify the signal from the original ligand. They are important for the rapid and/or widespread responses to stimuli. Common secondary messengers are cAMP and cGMP. So we've talked a little bit about ATP previously. ATP stands for acetylene triphosphate because there are 3 phosphate groups. When we remove a phosphate group, we're left with ADP, which is acetylene diphosphate, because there's 2 phosphates. Then if we remove one more phosphate, then there's only 1 phosphate group left, so we call it AMP for acetylene monophosphate. If 2 phosphate groups are removed from ATP at once, we can get cyclic acetylene monophosphate, or CAMP. Notice that this phosphate group is bound to the sugar molecule with 2 covalent bonds. CAMP can easily be transformed into AMP by breaking this extra bond. CAMP and the counterpart of cGMP are often secondary messengers. Again, the original ligand binds to the receptor protein, resulting in the phosphorylation of the G protein. ATP is modified into cAMP, and that triggers a cell response. Many cAMP molecules can be produced while the original ligand remains bound with the receptor protein. And this is how a secondary messenger can amplify the effects of a single ligand molecule.